Thank you very much, Carlos. Thank you to all the organizers for inviting me. So I'm Canadian, I'm from Ottawa, but I've been in France for a long time now. If you're interested in sort of like the less touristy, scenic part of Paris in the sort of southeast corner, the 13e arrondissement. So I took the liberty of changing the title of my talk a bit to adapt it more to a applied math physics group as opposed to a biology group. So like Ignacio, I'm a physicist. I apologize in advance for maybe my lack of rigor and absence of existence proofs and so forth. I try to work with experimentalists a lot. So straddling biology, theoretical physics, experimental physics. So it gets a bit messy. I hope you have a bit of tolerance for that. So the problem I'm going to talk today is related to how forces are transmitted along the cell membrane. And you'll see a bit later how it connects with the theme of this workshop on free boundary problems. Okay, so the context of the problem is here I'm showing you a cell. Doesn't really matter what kind of cell. And what you see here is a small bead, micrometer sized bead, that's covered with some sticky proteins. And the bead is approached to the outside of the cell. The cell membrane is enveloped by a membrane, which is, you can think of as a two-dimensional fluid, more or less. And the bead is going to be held in an optical trap. Okay, so the bead is dielectric. It gets held in this trap, and then the optical trap can be displaced to deform the cell membrane. And what happens actually is what you might not see with the naked eye. Remember, we're in phase contrast, pretty poor resolution, but a very thin tubular membrane is pulled out of the membrane. It's pulled out of this envelope. Maybe in the next movie you see it a bit better. The arrow is pointing to this thin tube, about 10 to 100 nanometers. You maybe can see it kind of fluctuating in this movie. It's kind of fluctuating. Oops, not there. Oops, gone too far. 1 second. Oh, I'm going the wrong way. That's why. I'm an idiot. Okay. So you maybe have to take my word for it, but there's this thin tether that's stuck to the one side to this bead, and the other, it's being extruded from the cell membrane. So we call that a tether, and these tethers are kind of useful. They're not just an experimental curiosity, but they actually have some physiological purpose. They were discovered in white blood cells. The tethers, as the white blood cells are passing through the vasculature, part of the membrane sticks to the lining of a vessel, and it's carried along with some flow, and these tethers are extracted, which allow the cell to cling on to the part of the body, the vasculature, where it wants to be. So they have some physiological significance. Today, I'm going to talk more about how tethers can be used to probe the mechanics of the cell membrane. Okay, so what we can imagine doing is pulling out one of these tethers, through a pre-existing tether, and then suddenly displacing the optical trap to rapidly increase the length of this tether. If you are familiar with rheology, think of this as like a step strain experiment. Okay, so this is just my cartoon. And once you suddenly pull on that tether, you can measure, because remember, we have this bead is held in an optical trap, and from the displacement of the center of bead from the middle of the trap, you can, assuming a harmonic force, you can measure the force that's needed to hold on to that tether. Okay, so that's what these data show. So time t equals 0 is the moment of where you rapidly extend the tether, and then you can see how the force decays over time. So it looks kind of banal. The question is, what can these relaxation curves possibly tell us about cell mechanics, biology? So I'm going to try to frame it maybe in a slightly bigger context, which is as follows. This is a movie of an embryo during development. Okay, so it's got there's about 1,000 cells there. And as the embryo is developing, each cell is deforming, moving, dividing, and during all those processes, the membrane has to dynamically adjust to, say, a volume constraint or an area constraint or whatever. So let's just zoom in, cartoon of a cell. And so during these sort of dynamic morphological processes, the membrane, which I've just as a cartoon represented in red, is sliding against its neighbors, sliding with respect to the intercellular contents. And so the question that arises is to understand how forces are transmitted along this membrane. Because it's important not just as a sort of a passive response, but what I'm trying to illustrate in this cartoon is that across this membrane, so this is inside the cell, this is outside the cell, there's lots of protein structures that kind of pass through the membrane that link the cell to its surrounding cells or surrounding environment. And that as the membrane slides, it gets deformed locally, that mechanical change locally can propagate along the membrane and will influence the behavior, the functioning of proteins that are some distance away. So it's generally important, but not known, how forces are really transmitted along this membrane. So this is what I'm going to try to claim here, is that these simple tether experiments can provide a window into how forces are transmitted in more complicated situations. Okay, and this is just sort of the usual token statement that, understanding how tension specifically is transmitted, it has a whole wide range of applications. It's involved in cell differentiation, cancer metastasis, viral infection, and so forth. Okay, so concretely, in experiments, in sort of controlled in vitro experiments, what is the state of the art in measuring how mechanical perturbations of the membrane are transmitted? So the idea is to use two tethers. So in the first slide, I showed a single tether being pulled. And what this cartoon illustrates is a so-called double tether experiment, whereby one tether, let's just call it tether 1, is perturbed. Say we apply some change in length protocol. We rapidly increase its length. And then a second tether, a little bit away, called a probe tether. I won't get into all the experimental details, but essentially the idea is by using the probe tether, because it contains some fluorescent lipids, and the change in fluorescence is essentially a readout of the change in the local tension near tether 2. So if I pull on tether 1 a little bit, and if transmission of the force will be detected as a change in the fluorescence in the lipids in tether 2. So what the experiments show is sort of kind of binary. That is, what this is telling us here. So in the blue data means that, let's say I pull on tether 1, I rapidly pull on it. The vertical axis is the fluorescence of lipids in tether 2. And what you see is that intensity does not change, which we interpret as that essentially tether 2 is insensitive to the changes going on in the vicinity of tether 1. And vice versa. If I now, just by reciprocity, if I tug on tether 2 and look at the response at tether 1, which is in the red data, you can see that the fluorescence at tether 1 doesn't change. So these kind of experiments where you have a membrane in a sort of native environment sitting on a complicated substrate, which is an actomyosin cortex, as Axel spoke about yesterday, essentially, there's essentially no tension response. And biologists are all excited by this because there's a lot of emerging work understanding to what extent can tension, which is modified at one position on the membrane, can it affect biological functions at some distance away? Okay, and just when I said binary, if you take another experiment where you do the same double tether experiment, but on a membrane without this cortex, which would, allegedly create some kind of frictional resistance. In fact, and then you do the same experiment, you see that as you solicit one tether, there's almost an immediate response of the other tether. So this data is supposed to show that in the absence, when you pull on one of these sort of bubble-shaped objects, tension is essentially being, a perturbation is traveling essentially very quickly. So that's what I mean by binary. To date, so there's a whole flurry of activity in the last 10 years, and it's still not really understood, how exactly, we don't really have a quantitative idea of how forces are transmitted along the membrane. We just have kind of sort of either or pictures. And what I was trying to do, I don't know if I'm successful, is to revisit the single tether experiments. And maybe I can provide some fresh light, some intuition that might give us a more quantitative understanding of what's lurking behind these results. Okay, so let's just go back to the, yes?
Is there a reason why you use fluorescence? I mean, they are in an optical trap. So
okay, sorry. The experiments, okay, good, very good question. I should have said, please feel free to interrupt me at any moment. In these experiments, actually, this is from this paper, Shi and company from 2018. They did not use optical traps. They used I guess they had beads that were held by micropipettes they used, but more recently, yeah. So that, yeah, and Axel, yeah.
How does this relate to this like membrane reservoirs on smaller scale? Is this...
Okay, I don't know if I can say anything intelligent about that. The idea is that, these membranes, they're not just, okay, they're rough because there's thermal fluctuations that create, but there's also more structured reservoirs. Again, in these tether pulling experiments, I don't know if the time scales are such that, I mean, to disassemble a caviole-based membrane reservoir, it takes several seconds to minutes. I'm not sure to what extent you're unforming and reforming reservoirs in these experiments, but that's the question that's been bothering me a long time. I don't really know. Okay, so yes. Sorry, I'm kind of losing my hearing.
You learn something new by exchanging the roles of tether 1 and tether 2.
Not really. I think it's just sort of a sanity check to make sure that there's nothing privileged about tether 1, saying that if I exchange the role of the sort of transmitter tether and the receiver tether, I get the same. It's just to show that there's nothing there's no.
So the membrane is indifferent to.
Yeah.
Okay.
That's the idea. Okay, so just moving on slightly. Okay, so let's just go back to the single tether experiments I showed at the very beginning, where you have a tether and you suddenly pull on one, or you quickly pull on, change its length, hold the length, and then look at the relaxation curve. So this is not a new experiment. This was started by Evan Evans in the 90s or early 2000s, Evan Evans being from British Columbia. And you see a kind of generic decay curve. And, the sort of immediate conclusion is, okay, well, this looks like an exponential decay, but that's completely unjustified. This is what these people do in this paper from about 15 years ago. They say, well, we have this what we see quite generically is a very fast decay at short times, and then a kind of slower petering off at longer times. You think, oh, that's a double. I'm just going to put a double exponential with a short time scale and a fast time scale. Okay, fine, but there's no reasonable reason to do that. And so my idea was, well, okay, can we really understand where this decay is coming from? And hopefully, as a physicist, are there some universal features hidden in this data that would allow us to maybe connect physically what's going on? Sarah? A bit louder, sorry.
The second time scale couldn't correspond to the caviole opening or something like that?
I don't think so. I mean, the second time scale, again, it's around 8 seconds. I think these protein decorated structures, they take I think they take longer than that. Okay, so yeah, so that this is sort of the goal, is can we decipher some physics from these sort of generic looking relaxation curves? And so here's my outline. I'm going to first give a short overview of some classic tether mechanics, by no means a new subject. That'll be more on the static side. I'll talk a bit about a diffusion model of tension to understand the dynamics following these tether extensions. I'll present some numerical results and some asymptotic analysis that allows us to get a simple quantitative handle maybe for what's going on. This asymptotic analysis I'll see is valid for very large tether extensions. We'll try to cure that for the case of finite extensions and then compare the model and the results and its predictions with data. Okay, so here we go. So a tether, this is, as Axel introduced yesterday, the mechanics of a membrane are described pretty well by the so-called Helfrich Hamiltonian or free energy, which contains a bending part. Here, H is just the mean curvature of the membrane, kappa being the bending stiffness, and, and a tension term, which in the original work by Helfrich in the early 70s, the tension was a little bit of a murky, nebulous quantity that I hope maybe try to shed some light on it, illuminate a bit. And so what a tether is, and this is work going back to work by Derigny, Jülicher, and Pro in the early 2000s. A tether, imagine you start with a flat membrane, a constant tension. So you think of tension, the memory lip is sort of a reservoir, and you apply a point force. What they found is that beyond some threshold force, that you get a tether forming, essentially a cylindrical solution to the Helfrich shape equation. So by shape equations, what I mean is that you take this Hamiltonian, this free energy, and you consider the first variation with respect to normal displacements of the membrane. Okay, so this is a non-linear solution relative to a flat membrane. So you have to go beyond the usual Monge gauge to see the emergence of this cylindrical solution. And it turns out there are nice relationships between the pulling force F, the equilibrium radius RT, okay? So the force and the radius are inversely related. And there's also a nice relation between the radius and the tension and bending stiffness. So this second relation we'll focus on, it's sort of Laplace's law in disguise. So when I say Laplace's law, normally if you had just a fluid interface, you have a relation between the difference in pressure between the inside and outside and the tension. Here, however, because the tether is part, it's essentially at equilibrium, the pressure inside the cell or in the vesicle is essentially uniform, and because the mother vesicle or cell from which the tether is pulled is very large, you can essentially assume that there's no difference in pressure between the inside and outside. So let's neglect the pressure part of Laplace's law, and the atypical contribution to Laplace's law is the effect of the bending energy. And so what this is just saying is that you have a normal force balance, you have tension, which wants to reduce area, and you have bending, which wants to avoid large curvature. So they're in conflict with each other, and at equilibrium, you get this nice relation. Okay, yeah, so the little asterisk means we neglect pressure inside the tether. You can drive this assuming everything's nice in equilibrium. It also holds at local equilibrium. And what I mean by local equilibrium is we can define the tension thermodynamically like we do with a 3D fluid as we do the pressure. You can think of the tension as a negative of a two-dimensional pressure. Okay, so this relation, if we define the tension thermodynamically, even if, say, so N refers to, say, the lipid number concentration. You can generalize this to multiple lipid species and even have mobile proteins that are, so you can have a multi-component system. Even if the tether is not at chemical equilibrium with its surroundings, as long as there's a separation between fast processes and slow processes, you can still define a local tension, and you will have this relation between the tether radius and that local tension. Provided that you can neglect certain dissipative forces. Like if the tether is growing really quickly, you're going to have hoop stresses, viscous hoop stresses that will intervene in Laplace's law. But I just want to say I'm not doing anything violent by saying that this is also valid out of equilibrium, and that out of thermal equilibrium. And that's going to be a key point in what's to come. Okay. So that's sort of tether statics. Now, what about the dynamical part of the problem? So as I said, we're going to imagine pulling a tether from the membrane. Remember, the tether is connected to the membrane. And so as you pull on the tether, imagine you just keep pulling, you don't stop. Well, eventually, the lipids in the surrounding quasi-flat membrane are going to flow into the tether. And that flow means that the membrane is flowing through essentially a very dense medium. You can think of all these little protein studs that are sticking through the membrane. These are like forming a kind of two-dimensional porous medium. So what that means is, as a physicist, now I'm going to really make abstraction of what we have. My membrane is represented as a flat, this is the membrane surrounding the tether. I'm going to represent it as a flat two-dimensional fluid. Okay, again, I know that's not true. A membrane really has some hidden projected area because of thermal fluctuations, but you can think of this as the sort of projected plane when you zoom out, okay? And this membrane is not freestanding. It's sitting on top of some substrate, whether it's the cortex inside the cell or some kind of glycocalyx that surrounds the outside of the cell. In any case, there's a substrate which acts as a kind of sink of momentum. Okay, so again, in the sort of overdamped low Reynolds limit, if I consider a little element of the membrane and consider the forces on the membrane, well, what you're going to have is the gradient of the tension, which you think of this as minus grad P in a way. Viscous stress, remember that the membrane essentially, we're assuming the membrane is in a fluid phase. It's a two-dimensional fluid. And then some exchange of momentum with the substrate, where psi represents some friction coefficient, okay? Now, you can see that we have two terms that are linear in V, from which we can construct a characteristic length. We can show that this length, scale L, is essentially, if we have this toy vision of the membrane and the substrate as a kind of two-dimensional porous medium, you can think of L as, roughly speaking, the typical pore size, which is on the order of a nanometer. But we're going to be interested in dynamics behavior on a much bigger scale, like 100 nanometers, micrometers. So on the scales that we're interested in, we can neglect the sort of Brinkman term in the equation, and we have just a Darcy relation between the local membrane flow and the gradient of the tension. Okay, so that's a kind of key point. Now, again, as the membrane's flowing, we're going to neglect any exchange with the surroundings. Of course, on long time scales, I mean, these lipids, they can, depending on what the nature of their surroundings is, at some long time scale, they're going to be in chemical equilibrium. But we're going to neglect any exchange, so lipids are conserved, so we have mass continuity. And then the key point, well, a key point, is that even though we're in a dynamic situation, we're looking at a membrane that's flowing, again, the local equilibrium hypothesis comes to the rescue, where we assume that we still have a local equation of state that connects the tension and changes in the area of a given patch. Just think of it as the projected area. Okay, so this is the definition of what I could call E is sort of a stretch modulus. Think of this as just the two-dimensional analog of the usual definition of bulk modulus in thermodynamics. Okay, and so using the fact that sigma is an intensive function, the usual thermodynamic rigmarole, you can express the stretch modulus in terms of changes in the tension due to a change in the density of lipids. Again, little n is the density, projected density of lipids. Okay, so combine these three features, and what do you get is, not surprisingly, that tension perturbations relax diffusively, okay? They relax diffusively with a diffusion coefficient that depends on the ratio of the stretch modulus to this friction coefficient. Okay, so this is very distinct from, say, single particle diffusion. You know, you could imagine tracking a label, a fluorescent protein or lipid in the membrane, and you could look at its, you know, diffusive trajectory. This has nothing to do with that. This is more of a cooperative diffusion that pertains to tension. Okay, again, I'm not taking credit. This is not my, you know, I wasn't the first person to develop this. This was expounded on in this paper from 2018, which they did not cite, but this actually goes back to earlier work by Evans and Young in the 90s. Okay, so this is going to be sort of the basic PDE. Doesn't look, you know, pretty standard, linear PDE diffusion. What could be interesting? Here's the problem set up from a sort of physics point of view, is that we imagine we start out with one of these tethers. I should call this L, now that should be L0, L naught, okay, with an initial length and initial radius. Remember that if the tether is if it's static, we're going to assume that the tension is equilibrated between along the tether and in the surrounding membrane. So the initial tension everywhere, for simplicity, we can just call that sigma 0. Okay, so we have this nice relation between the initial radius and the initial tension. Now, experimentally, we're going to suddenly increase the length of the tether by a factor lambda. Okay? Now, the assumption is that if we pull on the tether rapidly, the lipids in the surrounding membrane cannot adjust, so we're going to essentially be pulling the tether at constant area because the surrounding lipids are essentially frozen. They're frozen in a way. Okay, so if the length increases by lambda, that means, because we have a roughly cylindrical geometry, the radius decreases by 1 over lambda. But now, remember a few slides back, I showed that even out of equilibrium, modulo some assumptions that we can justify, we have this, always have this relation between the tether radius and the tension along the tether, the square root relation. So that means that if the tether radius is decreasing by a factor of lambda, the tension there is increasing by a factor of lambda squared. Okay? Meanwhile, the tension in the surrounding tether is completely unconscious of what just happened near the tether and still has its initial value sigma 0. So we have a kind of tension shock, like a thermal shock a bit, okay? And so we have this sort of very, very sharp initial condition on the tension. We want to see how that initial condition evolves. We're going to solve the diffusion equation in the membrane surrounding the tether, okay? For radial positions greater than RT. But RT is evolving in time, okay? It's not going to be just stuck at its initial value. Because as lipids do flow into the tether, remember, we're holding the length. We have this bead connected to an optical trap. We're holding the length, so the tether is going to slowly swell. Okay, so what are our boundary conditions? Well, I would say, you know, far away, we have some sigma 0. And the problem what makes the problem a little challenging is the boundary conditions at the position of the tether. So at the position of the tether, we know that the lipid influx, represented by these black arrows, is equal to the change in area of the tether. So we're holding the length constant. That means that this gives us a relation for the rate of change of the tether radius because of lipid influx. Of course, we don't know what RT is. We don't know, but so we need to sort of close the system of equations, and what closes it is this, I call it SC, self-consistency. I don't know if that's the correctly chosen terminology, but we know that at every instant, we must have this mechanical relation between the tether, the instantaneous tether radius, and the value of sigma obtained by solving the PDE, the value of sigma evaluated at RT. Okay, so we have a problem where the PDE is linear, a free boundary problem. Okay, so I think that's why this talk maybe fits in with the theme of the workshop, but with nonlinear conditions that determine RT. Okay, so let's see how we can make progress on this system of equations. So numerically, we can solve it using a standard approach for Stefan type problems. And if you can, what these curves show is the tension as a function of time at different positions measured from the center, from the axis of the tether. Okay, so it looks like standard diffusion. If you put yourself at, say, some distance, I don't know, a micrometer from the tether, what you'll see at first is you have no response. Then you're going to see, the tension there is going to locally increase. And then as the diffusion front is passed, then it goes back to its initial value. And so if you track, it's not really maybe a useful observable, but if you track how the maximum, if you consider one of these curves and at what time is this curve a maximum, and you track where that maximum, how that moves, you'll see that it follows the usual square root that you would expect for any diffusive process, okay? However, so, you might be tempted to say, this is a two-dimensional diffusion problem, so being in 2 dimensions, the diffusive profile should just be 1 over t exponential minus r squared over 4 dt. However, if we look at the value, so if we substitute, say, r equals root dt, then if it was classic 2-dimensional diffusion, the maximum value of the tension would just decay as 1 over t. But that's not what happens here. The maximum tension, so these colors are for different extension factors. With increasing extension, the tension does not decay as 1 over t, but as some smaller power of t. But definitely looks like a power law, and so that, we get this seems to converge to around 2/3 for large extensions. So I think we have to abandon that it's just a simple diffusion problem and maybe think of something a little more clever. And so seeing a power law suggests, maybe there's some underlying self-similarity in the tension relaxation. So let's just, to clarify things, we'll just rescale variables in the following way. So important thing is that R0 is the initial tether radius before we extend. The characteristic time is just the diffusive time associated with that length. Then our boundary conditions, remember there's a flux boundary condition, it gets transformed in the following way. And so we have 2 important dimensionless parameters. One's obvious, just the extension factor, which can be big-ish. And the other is this quantity B, which I would call the tether capacity by analogy with heat capacity, in the sense that B tells you for a given gradient in tension, remember, it's the gradients in tension that cause the flow into the tether. For a given gradient, B tells you how much the radius is going to change for a given flux. So we'll just call that tether capacity. And what we see from the numerics is that for given extension factors and with increasing lambda, the tether radius seems to grow as a power law. Okay? So remember, if I'm pulling if I make a big tether extension initially, so it means it's very long, like conservation of area, the initial radius is very small. So it starts out very small. And if this capacity, which we can estimate from numerical numbers, if this parameter B is small, that means that the tether radius is going to change quite slowly. The lipids are flowing in quite slowly, and the radius is going to swell really slowly with time. And so what we see is that there's going to be sort of a time window, I'll call it delta TSS, over which the radius grows as some power law that's insensitive sort of to the terminal state R0. Remember, R0 is the initial radius. If we wait long enough, the whole thing should come back to equilibrium, in which the tension is just going to be against sigma 0 everywhere, and the radius should have its initial value prior to sudden extension. So if the lambda is very large, we were starting out with a very small radius, and we're going to grow slowly. And as long as we're far away from that red line, we can anticipate that there's going to be some scale invariance and hence self-similarity in the tension dynamics. Okay, I think with this crowd, usually sometimes I do this for a biology crowd. I don't think I need to talk about self-similarity. So how can we maybe go a bit further analytically with that, well, what we can do is, remember what we're trying to solve is the diffusion equation 2D, okay? Nothing too fancy. We're going to posit that the tension has this kind of self-similar structure. We don't know what the function g is. We don't know what the exponents alpha and beta are. And we're also going to assume, again, hopefully valid for large extension factors, that the radius, tether radius, is swelling with some power law. And we're going to anticipate and then verify that this exponent delta is less than a half. And what's special about a half? Well, that's not surprising, because when you plug in this ansatz into the PDE, you're just going to find, by balancing terms, you're going to find that it's just classic diffusion for, in terms of this scaling variable, R over T beta. Okay, so that's not a big surprise. What about the other exponents? Okay, so this is a bit tricky. I'm going to sidestep a lot of the ugly details. The idea, so if we want to find delta and alpha, assuming that delta is less than 1/2, that means that this rescaled variable u, R over T, near the tether, u is going to be very small. Because if, let's say, the tether is growing, with the power of 1 over 10, that on the scale measured in units of u, u is going to be very small. So we can actually find g, because if we're near the tether, that means the diffusion front has long gone. We're essentially in an incompressible membrane limit. Okay, that's what fast diffusion means, that any change in density propagates very quickly. So we're essentially incompressible. And we know that in 2 dimensions, incompressible flow is like 1 over R. That means the corresponding tension is in log R, or equivalently in log U. So we know that we can find this function G for U small. And from that, and the boundary condition, remember we have a flux boundary condition and we have that mechanical relation. From those two relations, skipping details, we can find that the exponent alpha is 2/3 and delta is 1/3, which is consistent with our assumption that it's going to be less than 1/2. Okay, what about this sort of generalized velocity A, this parameter that sits in front of R? Well, what we're going to do, again, we can plug in our ansatz into the PDE. We can solve this equation. Okay, I wouldn't know how to do it. I mean, it's just a linear equation, but you can solve in terms of Meyer G functions. I mean, Mathematica did it for me, of course. And then you can look at the sort of asymptotic expansion for small u, match it up with this, just do the matching, and you can find A has this kind of ugly form. I'm sweeping something under the rug, which is that A actually depends logarithmically on time, which might seem like bad news, but fortunately, over the time ranges of interest, like that this log factor is changing it's essentially constant. Okay, so if you're, again, maybe this is full disclosure, this is where things are not very rigorous. You can attack me if you want, but strictly speaking, this kind of breaks the self-similar structure, but let's not worry about too much. Let's move on. Okay, again, just going back to the numerics, what we can plot is, you imagine we pull on the tether a factor, we increase its length by a factor of 16. Then you can plot the tension as a function of position at different times, and you can see, as usual, diffusion, it's kind of flattening out. And then with our exponents, and we can plot the rescaled function, we see that these data, they collapse pretty nicely and follow this master curve, which is, again, some Meyer G function. So that seems to work out pretty well. The problem is, let me just check time, 10 minutes. Okay, I'll have to motor a little bit, that's fine. The problem is, so far, this asymptotic calculation assumes that the extensions are very large, okay? Very large means essentially the radius at time t equals 0 is 0, which of course is ridiculous. That's not true. Another way to say the same thing is that, remember, the force holding the tether depends inversely on the radius. So if the radius is super small at time t equals 0, that means the force is enormous. And that's patently untrue. We know that in these experiments, the tether force starts at some finite value and then decays. So how do we deal with that? Well, if you consider smaller extension factors, what's going to become more visible is there's actually a first quote unquote self-similar regime where you do not have this growth law of the radius, where instead, remember, if I pull on just a little bit, say by a factor of 2, the tension is going to increase at time t equals 0. Remember, there's this lambda squared. It's going to increase by a factor of 4. And what happens is, so this is in this rescaled, there's a sigma minus 1. So it's kind of pinned at 4 or 3. And because that tether capacity B, which is telling us how fast the radius can change, because that's small, essentially this initial sharp profile is flattening out, but keeping the tension roughly constant at the tether. So that, again, I'm sliding over a lot of details, but that's going to imply that there's a slightly different dynamics at short times as compared to what I would call intermediate times, which is what I've spoken about already. So roughly speaking, at short times, at t equals 0, in fact, the radius is not 0. It's 1 over the extension factor. And we can show quite readily that at short times, the radius actually not growing with the power 1/3, but linearly. And what I'm plotting here, this is from the numerics. It turns out that it's convenient to plot this sort of rescaled radius lambda RT minus 1, because in that way, at RT equals 0, at time t equals 0, this quantity is 0. And what we see is, for different extension factors and different values of the parameter B, is that all these curves seem to lie on a common master curve. So there's some kind of underlying homogeneity in the problem. And where these data seem to interpolate at short times from this linear behavior to, at longer times, this 1/3 behavior. And we can capture that with an interpolation formula that's sort of just motivated by 2 resistors in parallel. So you have a crossover from the linear to the 1/3. And what we're going to do is take this interpolation formula and try to fit the experimental data we have, or that I've gotten from the literature. Okay, so the main prediction, to summarize so far, is that once you suddenly pull one of these tethers and look at the force decay, it's not going to be exponential, it's not going to be double exponential. The key point is that the tether radius is changing in time. And that's going to lead to this, at least suggest that there's going to be a crossover from a linear behavior to a 1/3 behavior. So we take the raw force data, we want to transform it to make it look like the tether radius. So we'll just take the inverse and subtract off the initial value. And this is the same data, I forgot the references. These are tethers pulled from breast cancer cells. And okay, to the naked eye, it might seem on log that there maybe is a linear regime. And just to guide the eye, but maybe also to bias the audience, that it does seem visually consistent with the idea that there might be 2 regimes. Again, that just might be a confirmation bias on my part. Similarly, with tethers pulled from axons, so axons, remember these long extensions from neurons. Roughly speaking, again, I might be deluding myself. It does seem that there is sort of a 2 linear straight line regime. Let's now fit the data. Now, one subtlety in the data is that, remember, in this asymptotic analysis, I'm assuming that we're at timescales far from the terminal relaxation. Otherwise, the whole self-similar idea goes out the window. Okay, once if I wait long enough, and then what I'll see is that the data are going to depart from the 1/3 prediction, and they start to saturate because the system is just returned to its initial state. So we need to prune out these terminal points. Okay, so because we know at really long times this is not valid. RT is going to tend to a constant. Okay, so we need to. I'm going to skip over the detail, but we use least squares fitting, where we treat the number of data points as a fit parameter in the least squares. And what we can find is represents the chi-squared per degree of freedom. And what we're going to do is we're going to look at the minima in chi-squared and then find the corresponding value of the exponent. That's what we're after here, is we want to have an estimate of this scaling of this power law delta. Okay, and so at that, we're going to do is from the minimum, we're going to look at point TC. TC is essentially the number of data points we've included in our fit. We're going to look at, well, what is the corresponding value of delta? And we see that it's not too far from the red line, which is the prediction of 1/3. Okay. We can do the same from a bunch of other data sets, from these MCF7 cells and from neurons. What we see are overall, some distribution of points, of values that are a little bit underneath the prediction of 1/3. And I think one possible reason why the experimental estimate of this exponent is less than the prediction is because experimentally, the extension factors are not enormous. So it might be a bit of a stretch to say, are we really, at least the experiments that I have access to from the literature, are we really justified to use this asymptotic approach? And you can see that instead for lambda equals 2, these curves, the data, they don't follow this nice master curve, but they start to deviate downward because they're guided, because they're going to relax to the final value of 1. And that's probably is that the fact that even if we cut out the sort of long time data points, we're still influenced by the terminal relaxation, which is going to mean we're going to have a lower value of the power of the exponent than the predicted value of 1/3. Okay, so I will conclude now. So what I'm trying to convince you is that in these tether point experiments can shed light on how forces are transmitted in cells. Okay, this is a very simple, in a way, simple experiment that you just pull on tether, you measure the force decay. You should not blindly just fit with a single or double exponential. I've tried to provide some evidence that this model is consistent with experiment. It would be nice. I mean, I don't think they're experimentalists here, but if experimentalists did pull these tethers with larger extensions, I would expect that it would agree better with my model. And last thing, I talked a bit about these double tether experiments where you pull on one tether and try to detect the response at some distance away. And I said this is kind of binary. These experiments show essentially no tension propagation. And maybe this is just very, okay, I'm not saying provocative, but just as a conjecture, one reason could be is that I show you this data where at some time, at roughly some time range, you see a departure from the self-similar behavior and you see the influence of the terminal relaxation state. And that, if you take that time and you calculate from this scaling law, you say, well, this is happening when my 1/3, when the RT determined by the sort of intermediate time power law, when it becomes close to 1 in these reduced units. And that tells you, I go back to real dimension full units, it tells you how far the diffusion front has propagated up to this time. Because beyond that time, essentially your tension signal has been extinguished. Once the radius has gone back to its initial value, everything's homogeneous. And that gives you kind of a figure of interest. It tells you that this relation tells you that the diffusion front has traveled by a distance that's solely determined by the experimental protocol. That it depends on how long the length of tether you've pulled and the initial radius. Okay, I should stop there. I think I've gone a bit over time. Thank you for your attention. Any questions?